So, students, good evening. Finally, everyone, finally we are back. Welcome everyone, warm welcome to the another session of MBBT. We can say day 2 somehow. So I have divided actually initially what we used to have when I started these classes in July 2020 during the COVID time. So I made these lectures like 10 days lectures. So in each day I have like made this package of all presentations, all the PDFs, all the protocols, all the videos, all the virtual protocols, and each day is been divided into a specific topic. Yeah. Um, so it usually used to take 2 hours for me to do so each day, so around 20 hours. So I'm trying to do same in this class, which is actually not normal from the way it was told. Usually we used to have in a month 5 days a week class, 1 hour each day. That was making 20 hours. But now, as I told yesterday, I might have to travel around, so I need to might be finished by this Saturday or Sunday, or might I have to go tomorrow. I don't know. I have no idea. But the day I am here with you, the time I am with you, I will try my best to give you all classes, to give you all content without any stoppage, without any hindrance, and having a very nice live classes and with the excellent learning material also. So that is my goal with all of you. So, and the last night there were very few students have joined me. So, but I just want to let you know all that we all also have classes at 9 p.m. So please join me at 9 p.m. also from 9 to 10, and we will continue with the same lectures that we left before. So it's almost 5:10 now. So I will start the session by 5:10. Yeah. Hello, Akshay, Anshimita, Arun, Dev, Shipra, Aishwarya, Shanmuga, Momita, Sojit, Tanvi, Hema, Shinu, Sumran, Shruti, Pooja, two Poojas, Gopika, Asmita, Shrutika, Vaishnavi, Soumi, Asima. Yeah, welcome everyone. It's very wonderful actually to see like all the students coming from all over India and joining. It's not like from one university and having one specific students. So we are having from students from all over India. The name I have just said. So that's the beauty of this course that I always love in this teaching. So we are not going with a specific part, but something bigger and sovereign. Beautiful. So let's start the session today. So far we are done with the basics of molecular biology, and we have taken the course of tools in molecular biology, right? So our two things were over. Now today we're gonna discuss about light and electron microscopy. Yeah, how does the light and electron microscopy works? So regarding the same. Um, yeah, diversity but equality. So let's start with the very basics. Now, so I have shared these lectures already with you. So microscope basics. So as we know, these microscopes, their main objective is to make things looks bigger. And this way scientists could help us to study the living things which are too small, which cannot be seen with the naked eye. So this is the body tube, this is the ocular lens, this is the nose piece, arm, stage, coarse adjustment, fine adjustment, base, light, diaphragm, stage clips, objective, nose piece. So always carry a microscope with one hand from here and one hand under the base. So you might be all aware of this basics microscope. I remember from my class 11th, I have taken biology as subject in my class 11th. So we used to tend to go and see dicot stems, monocot stems of the rose petals, yeah, of the rose leaves actually, and other stems also. So it was very wonderful to see all the cell coming within the cell, and the green cells with the cell membrane and nucleus in between. So it was my first like chance to see the basics of plant cells. Then we were taught in class 12th about mitosis and meiosis, how at the metaphase all chromosomes are coming over this in the center of your what is this plate, center plate, anaphase, I don't know what was called when metaphase, yeah, the metaphase, where all your chromosomes come into the center, lie over the center part of the spindle fiber. So we were able to see those fixed things. So it was very wonderful to see them. And these things keep continuing. We keep finding more advanced in our masters. We tend to work on our cancer cells, and then in PhD we want to work on heart cells, cardiomyocytes. So basically this microscope is a thing which have been always been helpful, always been handy to identify the mechanisms that are going within the cell. Yeah. So all the drug discovery, all the pharmaceutical industry, all the biotechnology industry, everything is based on this microscope. If microscope has not been there, then it might be it might be very difficult to predict things, how things are happening, because we can do things biochemical pathways by doing some assays for photochemical assays or biochemical assays, but in order to see things what is happening in the cell, this is only the microscope could tell. So there are three types of microscope: simple, which has only one lens; compound, two sets of lenses which can magnify 100 to 200 times larger than they are; then electron microscope, which can magnify objects up to 3000 times, and they do not use lenses but use electrons to enlarge the image. Yeah. So today we will be discussing more about the light microscope and electron microscope. Under light microscope we will go many diverse type of it, and an electron also, your scanning electron microscope and transmission electron microscope. So parts of microscope. So it has ocular lens, that is your eyepiece, through which length of the microscope that you look through. Then comes your coarse adjustment, the large knob on the microscope that you turn to bring the object into focus. Then the fine adjustment, the small knob on the microscope that brings the image into the focus. Then parts of microscope. So it has arm, yeah, as we discussed, which is supporting the body tube. Then body tube, the part that holds the eyepiece and objective lenses. Then nose piece, the part of the bottom of the body that holds objective lenses and allows them to be turned. Then high power objective lens, which magnifies around 40x, and there is a low power objective lens which magnifies to 3x or 4x. Middle power objective lens, and they have the power of 10x and 20x. So all these three different lenses, your 3x, 10x, 40x, they're all coming within this one circle of your eyepiece, objective lens. Oops. Then comes the stage where you put your slide, then clips to hold it, then diaphragm, which gives the light to be enter into it. So this is the diaphragm here at the bottom. Then comes the light source at the bottom. Usually nowadays it's electron, like with the light it is there. In earlier times we used to keep mirror here, and there's the at the bottom base to support it. Yeah. So let's repeat again. Upper is the ocular lens, then comes body tube, then nose piece, objective, stage clips, diaphragm, light, arm, stage clips, coarse adjustment, fine adjustment, and the base. So this is the field of view in the area that you see when looking through the eyepiece. Yeah. So this is always opposite. Like if it is E is like that, then it will come opposite and other way around. Always in the opposite direction. If you put your object as E here like this, you will see your object upside down and opposite. So this is the magnification. This is 70x and 10x. So which of these images would viewed at higher power of magnification? So this one, like this is a melanoma of one cell, and you can see the same thing in magnified form in this way. So, yeah, meta faceplate. Thank you, Shalu Kataria. Thank you so much. Because there are so many things are going in mind, sometimes we tend to forget the basic part. Thank you very much. Good. So this is the how you can calculate the power of a microscope. You need to multiply the power of ocular lens by the power of the objective lens. So this is a 10x here, and this is 40x. Yeah. So what are the powers of magnification? So in order to find out the magnification of a lens, it is found on the side of the lens. Magnification power of a lens is always identified by the label of x, 10x, 1000x. So multiply the power of the eyepiece by the power of objective lens. Examples are eyepiece and objective lens. That is, eyepiece is 10x, objective is 100x. Then you multiply 10x with the 100x. So your magnification power, your power of the lens would be 1000x. 10x multiplied by 50x, 500x, and so on. So as I said, your appearance of the specimen is upside down and backward. So if it is like this, it will go upside down and backward also. Like this. So this is the practical experience, but we cannot have it at the moment. So how to mount your slide? Like if you want to check any specimens, first you put a drop of oil or water on the clean slide, then use tweezers to lay specimen on the drop of water, then gently touch the cover slip to the edge of the drop of water to cover the specimen and the water. How to make wet mount slide? So get a clean slide and cover slip from your teacher, then place one drop of water in the middle of the slide. Don't use too much of water, will run off the edge and make a mess. Then place the edge of the cover slip on one side of the water drop. So this is the water drop here, this is your cover slip, put at the 45 degree Celsius, 45 degree angle, and then lower it over that. So place the side on the stage and view it first with the red banded objective. Once you see the image, you can rotate the nose piece to view the slide with a different objective. So this is one cartoon. Here we have three pairs of jeans, three legs pairs of jeans, 1, 2, and 3, and with 3 pockets. This is the mutant jeans. And yeah, so just very I know this is very basics about microscopy, but the reason here is that once we know the basics about anything, then it's good to go in the advanced part. Yeah. So I see many times students are complaining to my seniors that sir is just teaching us basics, there's nothing advanced, we wasted our money, we need our money back, it's all waste of time, why I'm here, and blah blah blah. And I feel really like what should I don't have words for that, what I feel about that. But this thing I learned from Germany. that whenever you start any topic, whenever we start to learn anything, a basic is really must. Since our ABCD of one alphabet of a language is clear, only then we could go to the advanced grammar, and then we can start making sentences. First we make very simple sentences, then gradually there is a thing called vocabulary. You started to learn more complex words, and then you can start making complex sentences also. Yeah, so this is a step by step. Yeah, this is one step by step which I'm trying to incorporate into my teaching also. So first we go very basic. So that's why on the first day, very basic DNA, RNA, protein translation, transcription with videos, graphics, with the animations, all these things were clear. Now we have discussed at the moment with very basics about the light microscopy, that how a basic microscope that you use looks like. Now from here onwards, I will take you towards the advanced part, that is light and electron microscope now. Yeah, let's start the next session now with the light and electron microscope. So here we can see a replica of van Leeuwenhoek from 1632 to 1723 microscope constructed in 1670. Yeah, so this is the an old model, and same way this is the replica of Culpeper tripod microscope, very nicely with the golden plates with the light with the mirror at the bottom, and you can place your object here on this mirror and you can see your stuff. Very nice one, old ones. This is a replica of Marshall microscope by John Marshall, present at the University Texas Galveston. So they're still maintained very nicely. So this is the content of light microscope with the 7 different subtypes in that. That is your bright field, dark field, phase contrast, polarizing, DIC, epifluorescence, confocal laser scanning. Yeah. And then within these 7 types under the electron microscope, there are scanning and transmission microscope. And then under the specimen preparation, we have light microscopy, scanning electron microscopy, and transmission electron microscopy. How do we prepare specimens with regards to these different types of microscopy? So in general, we are having light, scanning electron, and transmission electron. SEM and TM is a short form for that. So that's the basic content that we're gonna deal today here. So as we have discussed that microscopes people are being using by the various cell biologists, molecular biologists for various general techniques preparing specimens. So this is the old molecular bright field microscope with fixed stage and mirror. This is the light microscope with the bright field microscope here that we have just discussed. This is the general being used in the lab with the 2 monocular eyepiece and a binocular eyepiece, sorry, and having 10x, 40x, 50x and 60x in it. So this is a binocular bright field microscope with movable stage, dioptric adjustment, condenser and iris diaphragm, built-in light source. So you can make light here turn on and make it happen. So the most common general use of microscope is a bright field microscope, are named because the microscope field is bright while the object being viewed is dark. So here is a simple design, light directed at specimen, Unstained specimens. Stained specimens show excellent contrast. Ideal for stained bacterial cells and tissues. Good resolutions, bright background, tungstens or halogen light source. So this is the image that we get out of the bright field images. So here we can see very nicely the light first of all is coming in the background. So the yellow light, red light, white light. We can see a lot of light there. That's why it's called bright field images. So this is a section of your cut of ciliated parasites. This is the stained blood cells in the peripheral blood smear, and this is the flagellate trichomonas, right? So this is how the images looks like differently, and we can see in the background it's pretty dark and looks like that. And the basic design of this bright field microscope has been modified for special uses here. So inverted microscope on the right hand side we can see is the viewing of cells in flask, well plates or other deep containers that do not fit between the objective stages of standard bright field microscope. Yeah, so inverted microscope positions of light source are being used. Here we can see that the light source is from the top and the objective lens is at the bottom, and here is the eyepiece here. So in this way you can this is the place where you can course adjust your slide. This is a 96 well plate is there, with each 96 wells are being over this nice plate, and in each well you can have different conditions and you can watch it yeah, with each cell part in it in detailed manner, what is happening in this section, what is happening in this section, and it's one of the easiest way to check it actually. And in my PhD I use mostly this inverted microscope to do all my projects, all my things in that, and it's one of the most common microscope are being used. If you want to check your cell morphology, if you want to count your cells, you want to check whether your dye has been worked out or not, so this is the highly advanced technique or method that has been used. Yeah, I hope you all had yeah, thank you Anshimita. Dr. Urvi Parmar, good evening, very good evening. um, yeah, Anshimita, that's true. It's always good to make that thing. I hope you also, some of your students have been microscope in your life. If you have been, just share your experience in this chat box, where and which place you have joined it, and what did you find out of that. Yeah? And if you're still using it, in which experiments, how your day-to-day life with that. If you have any problem in finding something, you can also ask here to do some troubleshooting that something is not working out, or in future wanna explore these microscope, share things here. Yeah, we'll come back and read your comments. So your light microscope is done here. Then comes your dark field microscope. So the dark field microscope, which creates a dark background to allow viewing of small unstained objects such as motile bacteria that would be difficult to view in the bright field. The central portion of the light is blocked so that only oblique light strikes the specimens. So this is from the 19th century. Here we can see this dark field microscope. We have a dark background. In the light microscope we were having light in the background. In this we have dark background. And light not scattered by the specimens bypasses. Can see very small objects, but resolution is variable. And high contrast, good for unstained, live and motile specimens. So this is the dark field images of the Leptospira, a spirochete viewed with the dark field microscopy. The algae Hydrodicton reticulum viewed with a dark field microscopy. Yeah, so here we can see there's a dark field in the background, and we can see your specimens very nicely. This stage, this hexagon shaped cells, pentagon shaped cells, and this is the spirochete with the S shape in this bead type. Then comes your phase contrast, which converts the differences in the refractive index in specimens to differences in the imaged brightness. So the central portion of the light source is blocked here, creating a ring of light from the condenser that illuminates a specimen. The light waves refracted but specimens are slowed by the phase retardation plate, increasing the difference in the wavelength between the refractive and unrefractive rays. So we have a technology, this from the 1940s, provides high contrast, good resolution, good for bacteria, flagella, cilia, organelles, good for unstained or live mounts, and phase halos occurs in them. So we are having these annular rings here in this phase contrast microscope, and we are having phase objectives here. That's the major difference, and the images looks like that. So this is the, on the right it's a DIC image. We will see what's that in later part, and but this is the main image that you get from the unstained squamous epithelial cells. Um, the inner cellular organelles within this cell with the nucleus here, we can see very nicely and nicely done here. All these, this makes sense. Then polarization microscope. This is the in the polarization microscope, we are having a polarizer on the top and at the bottom also, and the specimen is put in between. And we also get bright image, dark background, and could be used for actually crystals like from meteoroids coming from outer space in order to find some minerals. So this is very useful in that case. So this is the Martian meteoroid that came onto the Earth and viewed under the polarizing microscope. This is the top right view, the bright field. This is the bright field view of the same epithelial cells, and the bottom is the high magnification view of the same cells with the polarizing microscope. So in the light microscope, in the bright field microscope, it looks like that, but in this microscope, the image looks like this four base, this Maltese cross. It's called Maltese cross in the polarized light, and this is very nicely. It seems like a picture from the church. Yeah, in the church, you can see in the glass, there is a painting is being done on the glasses with a lead Madame Curie, not Madame Curie. Uh, the one who made Jesus born. Who was that lady? I have to remember her name. Who made Jesus alive? Who was Mary something? Mother of Jesus? Yeah, Mary Saint, na? So you see these images over the glasswares somewhere. Paintings in the church. Sorry, I'm going off the topic. I just want to compare that meteoroid image. Okay, I cannot find that image somehow. Yeah, like this. Yeah, this one. Yeah. So they used to make in the churches like that image, and I found the same somehow that image that way. Then comes your epifluorescence microscope, which allows detection of molecules and ions within the cell. This is a much more advanced. So as we are going from light microscope, then bright field microscope, then dark field microscope, then polarizing microscope, then we have discussed phase contrast microscope. Yeah, so each time we are going into the more advanced level. Yeah, they are giving a better image, better resolution, high contrast, and in this case we are using a light source as mercury or xenon arc lamp. Special fluorescent dyes used to locate, and black background, bright stained specimen, and multiple fluorescent probes are available and detect small quantities molecules. Can use antibody stain techniques also. So this is the image from the epifluorescent image, which having a dark background here. So this is from the tetrahymena, a cytated protozoan, using the fluorescent dye here. So here we use fluorescent isothiocyanate. Yeah, that's called FITS. Yeah, which emits this green light. So here we can see on the left is a cell stained with the anti-basal body, and above right is a stain with the antibody against cell membrane surface antigen. And we compare the information obtained using the fluorescent technique with the bright field of the tetrahymena stained with the hematoxylin. Then this is the epifluorescent image showing metaphase in the new lung cells. So three fluorescent probes which are specific for DNA, keratin and tubulin. So here we can see this, the blue one is showing the metaphase chromosomes, right? And the red one are the keratin filaments. And the yellow ones are the spindle apparatus made up of microtubules. So in this way we have three dyes and we made it into the cell. So see, this is actually, yeah, very nicely the dark background is there also, and we can see very nicely all these things at the metaphase plate being separated, and the spindle fiber are being making these cells to separate into two parts and creating a cell membrane then later on, and separating into two cell parts with a different number of chromosomes, yeah, same number of chromosomes but in two different parts. So very nice image, very nicely shown, which was actually initially was very difficult to identify them. Initially we used to find them with the in the light microscope that we tend to have in the school, just with one light, that your chromosomes are being separated. We can't see any spindle fibers, we can't see any microtubules, we are just seeing chromosomes, they are being making a metaphase plate, and that's all. But with the help of various dyes present in the market at the moment, fluorescent probes, you can play around very well. You can play around with many things very well actually. I want to show you, maybe if I have that thing with me. No, I cannot, I don't have that paper anymore. No worries. Yeah. Then comes your differential interference contrast, that is your, it has a combination of your previous two microscope that we have discussed. So it will has also mercury lamp as a UV source. It has a beam splitter, that is a Wollstone prism, and it also has polarizing filter, and it has a recombining prism. Yeah, so it will have all these parts here, uh, and it will give you even more advanced images that what we have discussed before. It will give you a 3D image, so you can see how differently this microscope looks like, so big and so broad and with so many options in it. So definitely we will get a very high contrast, high numerical aperture, excellent resolutions. We can good, it's very good for unstained specimens, live mounts, and it could detect also the various RI, refractive index of the specimens. So here we can see the image that of the cricket ovule, yeah, cricket, showing the oocytes here, um, with the mitochondrial clouds. So this is showing a 3D dimensional appearance. So bottom is the epifluorescent images here of Balbani bodies, which are stained with the rhodamine 123 fluorescent stain specific for the mitochondria. This is the nuclei, yeah, both. This is the oocyte. So this is a 3D image we can see here with the DIC. This is a normal image, yeah. This is the DIC image. This is the epifluorescent image. So we can see very nicely these Balbani bodies being present here and showing a very nice structures in them. And comes, this is one of the, actually in the lab, if you got a chance to work, if you got a chance to work with, you students have replied here, nice. Zoology lab in my bachelor's in dark using my mobile light to power failure of the whole city. Funny. Right from school till my bachelor's have been using microscope, mainly using bright field microscope only. Great. Class 12 for the first time, and during the BSc in microbiology. Well, Asima also have same experience. Those were fun days. I can understand that. Yeah, that's very strong point you all made here. Because of this COVID thing, the education system has drastically changed. Yeah, we cannot assess all these things practically. We cannot work in the lab as we were working previously. Everything seems to be um, compressed within the laptop and over your bench, and that's it. It's become a, yeah, scare. So that's very good point you made. Um, but nevertheless, you all people have survived this time. Yeah, you all had survived this one of the biggest hurdle of our lives, the two waves that India have seen, 2020 and 21, and we are all the lucky ones. I hope our near and dear ones are all fine. No one had any extreme things in that, and everything by the God grace is fine with all of you. Just keep going on. Something good is coming up. More opportunities are coming up. Since this COVID hit, the biotech and the allied life sciences subjects are gonna be more commonly. The more jobs will coming up. Yeah, in industry also, in the public sector also, in the private sector also. So, firstly, in life, I handle microscope in my BSc in SP College Zoology. Both and mono observed more than 30 experiments and hundreds of slides. Now, because of online, everything I'm missing. Yeah, very sad. Epifluorescence seen in the quantitative microscope. Yeah, you can. In the 11th, Plasmodium. Then I joined BSc in microbiology, see many other microbial world. And nowadays, I work in the pathology lab as microbiologist. Wow, that's great. That's great. Yeah, bache, these are in the ovarian, this crocal ovarian um, crickets of the ovarian. They're the Balbani bodies are present within the cells. Yeah, it's a part of their structure. So in pathology lab, I use bright, dark field as well as phase contrast microscope also. Wow, that's beautiful. That's great. So you are the Akshay. Even I have never used this microscope. You are the most advanced person here in our batch. So you know the most about this microscopy. If have anyone has any query about microscopy, ask Akshay Davra, who have used these things practically. In my life in personal, I have used mainly bright field microscope, inverted microscope, dark field microscope, and one confocal microscope. That's it. That's the four or five I have been used, not so many. And mostly you are as a microbiologist working in the pathology lab. So for you, this might be the uh, one of the important part to use various microscopes. From that's the thing. If you have any practical aspect that you are looking for in anything that you have wanted, so you can use according to that. Mhm. Just a second. My father is there. Let me see if he needs help for the moving the scooter. Just coming in a second. In a second. Yes, Carla. Okay, so yeah, uh, sorry, it was someone else. Yeah, Faces used to use 3D images. That's right. That's right. So let's continue with confocal laser scanning microscopy. So in this is actually it's more one of the advanced level and mostly done once you done your work in the bright field or on the previous microscope we have seen. Then at the end person goes for the confocal microscope, which gives you a 3D image, and we use a laser in that. That is a krypton or argon laser, which is a high resolution sharp image with very high sensitivity, can be used in the reflectance or fluorescence mode, which eliminates background interference. You have a Biorad confocal microscope showing fluorescent labeled cells on the monitor. So it's very one of the expensive actually. If there is a, I think in the Indian Institute of Sciences, um, all the ICER, yeah, all the IITs, where they have a bigger bioengineering and biochemical department, they might have these microscopes because the maintenance cost for the laser and everything for this microscope is pretty much expensive, very expensive. And only those students or the PI or the researchers works which are expert in that. Not anyone could use them actually. So an expertise is required while using confocal images. So, and above this is the bright field image of Trichomonas, which is a posterior flagellum, can be seen with the exostyle, which is unstained. And left image is optical sections of Trichomonas immunostained with antitubulin and fits. This is the fits is mostly used to identify things. So here we can see the image of this Trichomonas posterior flagellum being present like that. But this one you having much different level. Yeah, you have posterior AF and C. We can see very nice images, 3D images with a dark background, and we can predict many things out of it. So in my case, when I was doing my project, so ठीक ठीक रख दे रहा. So I made a live microscopy. That was my PhD thesis. Redox regulation of PP1 and ER stress regulation of connective tissue growth factor in cardiomyocytes. And here I have discussed the, here we can see 4 different images here. Yeah, this is the control. This is 100 micromolar of H2O2. This is 1000 and this is 10,000. Yeah. So in the control, it seems things to be like working in this way of cardiomyocytes. At 100 micromolar, things were almost similar. At 100 micromolar, it starts to get more differentiated. At 10,000, even more. So it starts to destroy things. So this was actually run for 2 hours, but I made it into fast and I can watch them for like 5 seconds or so. And then we use some MATLAB data to identify the contraction of these cell lines. So that's how the microscopes are so useful to identify your results. Yeah. Now comes your electron microscopy, that is your scanning electron microscope and transmission electron microscope. Let's take one by one. So in the scanning electron microscope, you fix and dehydrate your specimens that you want to see, and there is stubs in that on which you fix on them, and then surface coated with the gold, palladium or rhodium is added, and the specimen is placed in a vacuum and electron beam scans back forth over it. The electrons that bounce off the metal coated specimen surface are collected and converted to a digital image and displayed on a TV like monitor. So electron beam is focused using a magnetic field in this case, and it gives a 3D image, also the external topography of the specimens, and very much higher resolutions and magnifications than all the light microscope we have discussed. So here we can see on the left side the DIC images. Yeah, this is the DIC images. On the top we can see the SEM images. On the left is the SEM of trichomonas, and on the right is the tetrahymena, and the bottom is the DIC images of the same. So we can see a 3D image actually very nicely, all these follicles being present very nicely here, and here we can see also very bright and very nice images of your tetrahymena. Yeah, so that's the advanced part of it. You get a 3D image, and the transmission, it gives a 2D image, but it also reveals the internal structures, internal organelles, yeah, with much higher resolution and magnification. And here also the electron beam is focused by your magnetic field. So see, on the left hand side is the SEM image of your trichomonas. On the right hand side is the TM image, your transmission electron microscope. So here we can't see the internal organelles, but in the right hand side it's all possible. Everything is being cleared here. So we can see everything very nicely in this case. And yeah, so note that SEM details of external structures are visible while in TM are revealed. So you have this Golgi, yeah, this is hydronomas here, and flagellum, right, and then recombinant flagellum here, F, and then this cross sections of basal bodies of 3 anterior flagella here. So we can see all the interior organelles. Same way we can see any cells with all the Golgi body, mitochondria, centrioles, plastids, nucleus, chromosomes, in a very detailed manner, which might be not possible with all the microscope techniques that we have discussed so far. This is further the TM images. On the left is the rat liver cell, and the right is the pancreatic acinar cells. So revealing internal cell structures having nucleus, nucellus, yeah, rough endoplasmic reticulum, cell boundary, zymogen granules, mitochondria, and so on. So we have discussed all the basics with the example of all these microscope, how they are working, how the corresponding specimens looks like when you see under them, which is more advanced, which is more helpful if you got a chance to work with this microscopy. Beyond that, before you start your microscopy, there is a specific way, there is specific method before to make the specimen preparation, to make them ready for these different microscopy. That is your light microscopy, scanning electron microscopy, and your transmission electron microscopy. So this is the general schematic for preparing specimens for light microscopy. On the left is the live mounts with a view, vital stain and view, and on the right hand side, which is the fixed specimens, such as you first fix your cells, then dehydrate, infiltrate and embed sections, mount, stain, and view under them. Then live mounts, so to view organisms, tissues or cells in as close to the natural state as possible. Unstained live mounts are used. Viewing time of live mount is limited, and unstained specimens have low contrast requirement. And then supravital stains may be applied to provide more contrast or identify certain components, and these are stains that are not harmful to living cells. Then fixed specimens for histology and the study of tissue. The specimens may be preserved using chemicals such as formalin, acetic acid, ethanol, and methanol. So then you fix your specimens and dehydrate it by serial transfer through an ascending alcohol series to 100% alcohol. The specimens are infiltrated with melted paraffin, paraffin substitutes, plastics, and placed in mold to harden it. Then specimens are cut into 5 to 10 micrometer thick sections using a steel knife or razor on an instrument called microtome. Then vibrotome uses a vibrating knife to cut sections. Then you cut sections are then mounted on the slides. Then we achieve the contrast, and then we under the microscope for the same. And this is for the scanning electron microscope, where you fix your samples, then dehydrate, then mount on the stubs, then sputter coat and observe it. So first is fixation. So you use a glutaraldehyde, paraformaldehyde, osmium tetroxide, and after fixation you dehydrate with the help of ascending alcohol series to 100%. Then in the organic solvents such as acetone or propylene oxide, and the specimens for SAM may also be processed in critical point drying apparatus. The specimens are mounted on aluminum stubs using sticky tapes. Yeah, applying specimens to stubs arranged in a holder. So you can use this as one of example of stub. So you put over these stubs there, and then you put your sample on them, and then you check over the microscope. So you put your specimen inside this. This is the electron column which will hit, and the image will be displayed over this scanned images over this computer, over the computer screen. So it's being a bit huge, your SEMs, scanned electron microscope. Yeah, and then on the other side, the transmission electron microscope, you fix, dehydrate again, infiltrate, section it, apply sections to grid, stain, and observe it later on. So you need to fix specimens that are fixed in glutaraldehyde or paraformaldehyde, and then glutaraldehyde mixtures followed by osmium tetroxide. Then dehydration is accomplished by carrying the specimens through an ascending alcohol series to 100% alcohol, then to an organic solvent such as acetone or propylene oxide. Then specimens are then infiltrated with epoxy or plastic resins placed in the plastic molds to harden it. Then instrument called an ultramicrotome is used to section the specimen. So you have your specimens holder here and knife holder here, and then you can have a cutting of a sample from there. So in this way you have a glass or diamond knives which are used to cut the ultra thin section. So you have these kind of sections coming up with the help of this microtome. Then you put these sections over this tiny metal grid which are supported, and then you can put your samples over that, and then stains such as uranyl acetate and lead citrate are applied to make certain structures electron dense. So this is how the grids look like. You sit there, your specimens are inserted. This is electron column with additional image display and the phosphorescent plate for viewing the image here. So this was all about the light and electron microscope. Now I want to touch with you students a little bit about how we do use microtome. This is a bit old one to show, but also quite important. How a microtome really works. Sorry student, just take 5 minutes of yours.
Assuming you've already harvested and embedded your tissue samples in paraffin wax, obtain your paraffin cartridge from the refrigerator. Next, uncover the microtome. Next to this machine, you should find the slide former. Plug this in and adjust the dial to the middle position. Place a beaker of warm water on the slide warmer and keep a transfer pipette within reach. Next, remove a blank slide from the box and label it with your initials, organ type, and date. Place the slide down on the warmer and using a transfer pipette, put a few drops of water onto the slide. This series of steps involves paraffin cartridge preparation. Remove excess wax from the cartridge using a razor blade. Make sure to cut it in a trapezoid shape with the longest edge at the bottom of the cartridge. Be careful not to crack the paraffin. When you are ready to section the paraffin-embedded organ, place the paraffin cartridge in the microtome while pulling the main handle towards you. Use the knob on the right to adjust the section thickness required for your sectioning, approximately 10 microns. Next, adjust the stage with the wheel on the left as to move the paraffin cartridge close to the microtome blade. Unlock the wheel on the right by pushing the pin button down. Next, place both hands on either side of the finger guard and pull it down as to reveal the microtome's blade. Remember to always lock the wheel into place when you are not sectioning. As you begin to cut into the paraffin specimen, discard all unwanted sections using a paintbrush. Be careful not to drag the brush across the blade. Continue turning the wheel until you obtain a desired section. Once you have done so, make a ribbon that contains multiple sections of your tissue. When you are finished using